This method can help answer key questions in the PCSK9 biochemistry field, such as which drugs or genetic changes affect PCSK9's proteolytic function. The main advantage of this technique is that it allows a rapid and amplifiable readout of an intrinsically low-frequency biochemical event. The implications of this technique extend towards atherosclerotic heart disease, because PCSK9 proteolytic function is required for its ultimate effect on serum cholesterol levels. Though this method can provide insight into PCSK9 itself, it can also be applied to other biochemical systems, such as other proteases with intrinsically low proteolytic activity.
To begin, aspirate medium from HEK293 T cells. Wash with PBS and treat with the minimal volume of 0.5% trypsin EDTA. And incubate at 37 degrees Celsius for 2 to 5 minutes to dissociate the cells from the parent flask. Add 2 volumes of DMEM supplemented with 10% FBS to inactivate the trypsin EDTA. Then pipette the liquid in the flask up and down several times, dislodging the adherent cells, and transfer the cells to a sterile tube. Next, remove a small aliquot of cells. Stain with an equal volume of trypan blue, and transfer the stained cells to a slide, and use an automated cell counter to count the cells. Centrifuge the tube at 500 times G for 5 minutes.